Hi everyone, welcome back to Carl Chem after a bit of a hiatus. I'm Lainey, and today we're back with a new journal club. This article is a collaboration from a couple different universities in the UK, and it was published in Nature. The title is Mechanical Release of Homogeneous Proteins from Supramolecular Gels. I'm able to share this article with all of you because it's licensed under Creative Commons. So if you want to look it up yourself, the DOI link is in the description and you'll be able to access the article for free. The objective of this work is to show that supramolecular hydrogels can be a successful formulation for biologic drug product at up to 10% by weight, which can potentially be a substitute for cold drug storage. The rationale here is that cold storage is not environmentally friendly, it's expensive, and it's not available in low-resource countries. The current status of this field is that most biologics do require cold chain storage. Alternative polymer formulations are in development. However, the authors note that they often require some sort of stimulus to release the protein from solution, which isn't always practical and doesn't always separate out the polymer matrix from the drug, meaning that the polymer must be safe for patients and will require safety testing on its own. For this application, the authors are looking to encompass 5 major criteria. First of all, they want to reduce or remove cold chain requirements. This needs to be broadly applicable to many different drugs. It has to have 0 or very minimal excipients in the delivered formulation. It can't require complex triggers to release the gel matrix, and it needs to be able to tolerate high concentrations of biologics. For this, we mean up to 100 mg per ml. To get a general overview of this whole experimentation, I like to look at figure 1F and G. So what we have is the low molecular weight gel networks that can physically entrap proteins. And this is going to prevent irreversible aggregation because the proteins can't physically reach each other. The importance of preventing this aggregation is that it's a huge consideration in pharmaceutical formulations because it's often the reason that biologics will lose their activity. What they're actually doing with these drug products is that the gel with the encapsulated protein is formed in a syringe. This is how the material is going to be stored and shipped until it's ready to use. Then, when it's time to administer the drug, the gel plus the protein gets pushed through a syringe filter. The force of pushing it through the syringe will physically break down the gel matrix, which frees the encapsulated protein. Then the filter will separate out the gel in the drug, which is going to leave pure drug product for administration to patients. Now let's get more into the meat of the paper and talk about the gel formation. These gels are made out of low molecular weight gelators, which will self-assemble into long fibrous structures that form a 3D network. We can see the monomer in figure 1A and the gelled material in figure 1B. The special thing about these gels is that they're very stiff, but they'll break at low strain, which the authors are using here to their advantage. The authors show multiple options for low molecular weight gelators in the supplemental information, but ended up selecting the best one to showcase in this paper. To make the gels, a concentrated solution of low molecular weight gelator is combined with tris buffer at pH 8 to induce gelation. After the gelation occurs, the final pH of the self-supporting material is 6.8, which is great. It's just about neutral. Many drugs are formulated at this pH. When made this way, the gels are very flexible, so the authors added calcium salts to increase the elasticity. To get a first look at the physical properties of the gels, the samples are analyzed using rheometry, which measures the strain on a sample and gives insight onto the material's viscoelasticity, which is how the material will deform as a result of mechanical strain. There are 2 measurements shown in the rheometric data throughout this paper, which are G' and G double prime. If you're not familiar with them, G' is the storage modulus, which is representing the elastic or the solid behavior, while G double prime is the loss modulus, which is representing the viscous or more liquid state behavior. When you're interpreting these results, keep in mind that if your G' is greater than your G double prime, the material will be more elastic, so it's more likely to deform and bounce back to its original shape when you put it under pressure. Whereas if you have a G double prime greater than your G', the material will act more like a liquid, with pretty immediate breaking and not going back to its original shape when you put pressure on it. Looking at figures 2B and C, we can see the rheological data from the gels formed with calcium chloride, and these gels are encapsulating dextrin, which is a sugar. where the solid dots are G prime and the empty dots are G double prime, what we're seeing is that the G prime is greater than the G double prime, which means that the gels are able to withstand some stress and act more like a solid than like a liquid. Then the authors go on to discuss the shear strain, shown in figure 1C, where we see the G double prime in empty circles eventually becoming greater than G prime when put under strain. This means that with enough strain on the sample, the hydrogel will eventually break down, which will allow you to get the drug in solution without the gel matrix. To get an idea of what the 3D structure of these gels look like, the authors use small X-ray angle scattering and circular dichroism. Gels that include calcium chloride show a periodic arrangement with crystalline structure in the Sachs data because of this peak here. The CD data for the calcium chloride gels tells a little bit of a different story, and it indicates more of a beta sheet-like arrangement because of the negative band around 235 nanometers and the positive band around 190 nanometers. However, in the end, the authors note that the arrangement of the gel itself doesn't really matter. The important part is that the proteins will get encapsulated between the gel fibers, which makes them unable to move and therefore making aggregation impossible. Next up, the authors performed proof of concept experimentation using fluorescent dextran. We have a picture of this model in figure 2D where the authors show the gel plus fluorescent dextran payload in the syringe on the left side and then the filtrate on the right side. What you can see is that the product goes from opaque to clear after filtration. In this experiment, the authors see about 92% recovery of the dextran. Then the authors did NMR spectroscopy on the filtered material to check if any of the gel had gotten through the filter. And what we're seeing is just the CD005 gel in the black trace on the bottom and the dextran only in the blue trace just above it. What you can see is that there is some spectral overlap at about 3.25 to 3 and about 2.75. But overall, they do have two distinct profiles. There's also one additional control here, and that's the 0% by weight sample, which I think is just the gel matrix after filtration. And I'm seeing at around 3.25, we have some overlap here as well, which makes me think that we might have some gel component present in that sample, but it could also be something from the filter or the formulation as well. Moving on to the sample results, when you're looking at these results, what you're looking for is to check that the dextran peaks are present in the samples. If the dextran peaks are present, it means that you've successfully filtered it out. You also want to see an absence of the gel peaks, because if the gel peaks are absent, it means that the filter has successfully separated it from your intended drug solution. Now looking at the sample results from 0.1 through 10% by weight, all of these peaks present in the samples are also present in the dextran control and the 0% by weight control, which is why the authors are saying they don't have gel left in their sample after filtration. Personally, I'm not 100% buying these results because there is some spectral overlap in the gel trace, and I'd really like to see another analytical technique to complement these results. Then the authors performed FTIR experimentation on the gel portion after filtration, which is shown in supplemental figure S10. In that figure, what we're seeing is the filter only in part A, the filtered gel in part B, and the pure powdered gel in part C. And as you can see, the filtered gel is a combination of the filter trace and the gel trace, which shows that at least some gel is being retained in the filter. Keep in mind that this is not a quantitative technique, so you can't say how much of the gel has been retained, which means this technique doesn't prove that we have full gel retention in the filter, just that we have some gel retention. Then the next step the authors took was to incorporate a fluorescent group within the gel itself and measure fluorescence on the filtered sample. Results are shown in supplemental figure S11, and what they found was that a small amount of gelator was in fact present in recovered solutions, but it was at less than 1%. My only problem here is that when the authors performed this experiment, they used a modified gelator, and it really just leaves me wondering if this assessment is truly representative of their original gelator discussed throughout the rest of the paper. For the next step of this process, the authors did experiments using insulin as the encapsulated protein. Insulin is a really widely needed drug. It requires cold storage. It's very sensitive to agitation, so this is a great model for them to use. In this case, they made the gels without calcium chloride, and they prepared samples with and without an aggressive shake at 600 RPM. The corresponding control here is insulin that went through the same shake conditions and filtering, but with no gelator. Results in figure 3C found that shaking caused aggregation in the control sample, but no aggregation in the gelled sample. We also have the cell-based activity results in 3D, and they show that the insulin plus gel samples in blue have similar activity to their control of a fresh insulin in red, even after shaking at 600 RPM for 24 hours. To show that the gel is not retained after filtration for the insulin experiments, the authors used NMR spectroscopy again. This is done in a similar fashion as the previous NMR data that we looked at. The insulin-only control is on the bottom in black. The CD005 gel spectra is in blue on the next line up, and we do have spectral overlap again here. The authors are looking specifically at the peak occurring here at about 8, and they note that this is how they're determining if gel is present in the samples. Then we have the gel plus insulin control in green, which shows what a profile would look like if the gel made it through the filter. And then we have the 3.2 mg per ml insulin after filtration on the top in red. We aren't seeing the gel only peak in the insulin or in the filtered sample, which is why the authors are saying they don't think the gel is getting through the filter. I do have the same comment as before that the spectral overlap might indicate leftover gel in solution, and the authors do make a note saying that overlapping of peaks makes the analysis more difficult than for dextrin, but I'm most concerned with the huge peak in the filtered material trace because it's not in any of the other traces, and I don't see a note from the authors on it, so I'm not really sure what it is. If anyone here is an expert in NMR and has an idea what it is, please let me know in the comments. The authors also note that the rheological properties of the gel are impacted by adding the insulin to them. For the next leg of this paper, the authors also encapsulated beta-galactosidase with added calcium in the gel matrix. In this case, they performed a temperature assessment at 50 C, which is 122 degrees Fahrenheit, for 7 days. So this is high temperature for a long time. The authors found that the gel was still intact after this time, but the calcium addition appears to reduce yield. Then in figure 3F, we see relative activity results. The reference control is the fresh insulin, and the samples are the encapsulated beta-galactosidase at 50 C for 7 days, and the corresponding control to that, which is the beta-galactosidase with no gel at 50 C for 7 days. The gel encapsulated samples show around 85 to 110% activity to control. For an activity assay, I'd say that level of variability is comparable to control. The non-gelled beta-galactosidase solution that was held at 50 C for 7 days is only at about 30% of the control, and I'd say that this is a significant drop for an activity assay. So what we can say is that the samples encapsulated in the gels retain their activity, while the samples without gel did lose activity. The authors note that this performance is equal to another established hydrogel system, boronic acid PEG hydrogel. However, the CD005 gels in this paper can tolerate a biologically relevant volume of 500 microliters versus the 50 microliters in the other system. Then the authors show the activity data for beta-galactosidase gels with a 50 C hold over 4 weeks. We're seeing about 20% of activity is retained for the gel encapsulated samples, which is definitely impressive, but it also does show that there may be temperature limitations for this gel system after all. However, this would just mean storage at room temp, which would still be an improvement over refrigerated or frozen storage. For the finale, we have my favorite part of this paper, where the authors shipped the beta-galactosidase gels through the UK Postal Service and then tested activity. They share the temp tail in figure S26. We can see that the temperatures range from 17.6 to 24.2 degrees Celsius over the 3 days in transit. Then the activity data are reported in figure S27, where we see about 110 to 120% activity to the control. For an activity assay, 20% is pretty normal variability. I would agree that these samples have an activity comparable to control of fresh beta-gal, and I just love this experiment because it really is a real world experiment of what can happen to these samples over shipping. Now for my overall review of the paper, I thought this was a really great paper, and I particularly liked the way that it was written. I found it to be really easy to read, although you do need to have the paper on one half of your screen and the supplemental on the other half. For the science itself, these gels seem really cool, and I think that if they work, they can eliminate a lot of logistical challenges in drug storage and shipment, but this research is still very early. The number one thing for me is that I'm not 100% buying that the gel is completely filtered out because there was some spectral overlap on the NMR results, and there was a new peak in the insulin samples. I'd like to see a little bit more characterization on the filtrate here using a different type of test method to drive that point home. I think that some sort of chromatography would be a really nice complement, maybe like a reverse phase or a Hillick LCMS to separate that out, but it would really depend on how large your polymer fibers are. And in this vein, they should consider the safety of the CD005 gel fragments in humans. Depending on the safety and the route of administration, some leftover gel might be okay, but I understand that would be a large study in itself. I found it really interesting that the calcium chloride was not always necessary in the gels depending on what protein is encapsulated, and I think the authors should consider that in larger biologics there will be more surface charge that can potentially impact gelation here, meaning that they might have a lower dose cutoff for a larger or more charged drug. The last thing to consider is how are you going to treat your drug post-filtration? So what safety measures are you going to anticipate for the filtered material before it gets administered to patients? For example, how do you know the filtration was successful, or how do you know that the drug has not been damaged? All in all, I think it's a great paper, and I'm really looking forward to what these authors put out next. What do you think about this experiment? Let me know in the comments. And if you think my interpretation is wrong or if I missed anything, let me know about that in the comments too. And of course, like and subscribe for more.